Hi, my name's Audrey Bollas, and I'm a graduate student at The Ohio State University. I work at Nationwide Children's Hospital in both Elaine Martis and Peter White's lab at the Institute for Genomic Medicine. And my poster is titled A Machine Learning Approach to Detect Somatic Variants in Tumor RNA-Seq. So why do we want to do this? Firstly, reliably identifying variants from sequencing data is a crucial step in studying the relationship between genotype and cancer susceptibility in tumor genesis. And RNA sequencing is increasingly being used in a number of different analytical pipelines, such as to look at gene expression, allele-specific expression, RNA editing, and now it's also starting to emerge as a way to uncover variants as well. So the goals of this project is to create a pipeline to identify variants from tumor-only RNA sequencing data, and then to develop a machine learning model to classify these variants as either germline, somatic, or artifact, using the corresponding DNA variants as ground truth. Additionally, we want to uncover some biological insights by exploring the features that distinguish between germline versus somatic variants in our machine learning model. So the data was processed using almost entirely a SNAKE-make workflow, from the processing the BAM files all the way through variant filtering. Variants were then annotated using our in-house tool VARHouse, which contains information from external databases such as nomad allele frequencies, ClinVar significance values, and information from other databases such as effect prediction scores. Additionally, we calculated sequence context features, such as what were the surrounding context of the variant, and then also other variant properties were used, which we got from GTK, which is the software we used to call the variants. So looking at our results, this first plot shows the distribution of the labels for the machine learning model. So we can see there's just a ton of germline variants and a smaller amount of artifacts, and then a much smaller amount of somatic variants. And this is just the case biologically in our pediatric cancer samples. There's just not as many somatic variants. So we decided to actually build two separate models. So the first model predicted true variants versus artifact, using all of the somatic and germline variants as true variants. And this model performed pretty well. Specifically, the recall for the true variants was high, which is what we ideally would want to expect. Next, we took those true variants and built a model that separated out germline versus somatic. And this model didn't perform as well. The recall for the germline was really high, but for the somatic, it was not high. And this is mostly due to the fact that the data is so imbalanced. So moving forward, we're going to try to increase this model performance by trying to balance the data more before training the model. So next we want to look at some of the feature importance. So we used SHAT for this. So SHAT values summarize the feature importance for each model. So the first plot shows the true variant versus artifact model, and unsurprisingly, the alternate allele depth and percentage are most important for differentiating true variant versus artifact. To differentiate germline versus somatic, the quality by depth is the most important for differentiating these two. Additionally, whether the variant change was a transition also had a high importance. We can also look at feature dependence. So this dependency plot can provide insight on how features interact and affect the class label prediction. So in this case, it's the germline prediction. And we can see here that a moderate quality score reduces the predictive impact of the alternative percentage when distinguishing between germline and somatic. So these two features are actually highly interacting with each other. Next, we kind of wanted to do a quick comparison of the variants we find between the RNA sequencing and the whole exome sequencing results. So this Venn diagram just shows an overlap of these results. And so this does show that a majority of variants in the RNA-Seq do exist in the corresponding whole exome sequencing. However, there's quite a few variants that we aren't capturing from the RNA sequencing. So we did investigate this further. This plot on the right shows the variants in whole exome sequencing missed by RNA-Seq. So the darker blue dots are the total variants broken down by sample on the y-axis. And then if we actually reduce this number just by the coverage value, so if the variants in the RNA-Seq did not have a coverage of at least 10, there we can actually see much fewer variants. So this is the lighter blue dot here. So we think that a majority of the variants that aren't being called in the RNA-Seq that do exist in the whole exome sequencing is due to the expression amount in the RNA-Seq. So these variants just aren't being expressed high enough in order to call them. So some of the conclusions, SneakMake was used to design a pipeline to process raw data from RNA-Seq reads all the way from band processing to variant filtering in VCF format. Features that distinguish between germline, somatic, and artifact were uncovered using SHAP values. Our model, built with XGBoost, accurately classifies RNA-Seq variants as true variants versus artifact, and there is a high overlap between variants detected using RNA-Seq and whole exome sequencing. Some next steps, the main one is to increase model performance to more accurately classify germline versus somatic. And then next, this Venn diagram shows this small amount of variants that exists in the RNA and not in the whole exome sequencing. So this is kind of our next point of interest to study to see if we can find any interesting or meaningful variants in this case. So further investigate this small red area here. So look for potential RNA editing sites that would not exist in the DNA. And then also investigate allele-specific expression using RNA sequencing variants.